Morphological markers, refer to phenotypic traits that influence by environmental factors and by developmental stages of organism. So, close morphological markers represent gene tags. In addition, additive effects of morphological markers represent main effects of alleles, while dominance effects of morphological markers represent interaction between alleles, whereas linkage morphological markers represent by variance and covariance analysis between individuals. However, association morphological markers represent by calculating population mean. On the other hand, biochemical markers include allelic variant of enzymes, and these markers, like morphological markers, influence by environmental factors and by developmental stage of organism. Therefore, in Isozymes studies, one enzyme express more than one gene, while in Allozymes studies, one enzyme express one gene controlled by co-dominant alleles. In contrast, molecular markers, or DNA markers, occupy specific genomic positions within chromosomes to study genetic diversity between organisms. So, these markers, not influenced by environmental factors, and not influenced also by developmental stage of organism. Continuously. This slide referred to polymorphic and monomorphic bands of genetic markers on gel electrophoresis. As we show, polymorphic bands discriminate individuals, while monomorphic bands not discriminate individuals. Therefore, good molecular markers must be polymorphic and co-dominant inheritance throughout genome, with high reproducible and resolution. Hence, molecular markers use for genetic diversity and marker-assisted selection to get genome mapping. But be attention please. Single locus with two alleles, referred to biallelic markers, such as SNP markers. Continuously. DNA fingerprint. Amplify genomic segments that flanking by molecular markers by DNA extraction, plus markers amplifications, plus electrophoresis separation, plus bands visualization, plus band score, plus data analysis. This slide referred to examples of molecular markers. As we show, single locus markers include RFLP markers, SSR markers, and STS markers, while multi-locus markers include AFLP markers and RAPD markers. Finally, this slide presents flow chart from phenotypic level to DNA level. As we show, phenotypic level study biometrical model and additive and dominance components, while transmission level study Mendelian segregation and genetic relatedness, whereas population level study allele and genotype frequencies. On the other hand, DNA level study DNA structure, DNA organization, and DNA recombination.